All right, let's dive right into the microscopic world of our own bodies and talk about something absolutely fundamental, our cellular receptors. Just imagine your cells are these tiny, bustling fortresses. Well, these receptors, they're the super specialized gatekeepers, and they control literally everything that gets in and out. So, how in the world do these gates actually work?
You know, every single one of your cells is wrapped in this protective barrier, the cell membrane. It's an amazing defense system, but it creates a bit of a puzzle, right? How do all those critical messages, we're talking instructions from hormones, signals from your nerves, how do they get past the walls to deliver their orders? Well, the answer is our gatekeepers.
So at its core, a cellular receptor is basically a protein that's just waiting for a very, very specific signal. And that signal molecule, which we call a ligand, fits into its receptor perfectly. You've heard it before, but it's the best analogy. It's like a key fitting into a lock. When that key turns, boom, it unlocks a whole response inside the cell. It's this beautiful, simple interaction that runs pretty much everything in our bodies.
Now, this cellular fortress doesn't just have one kind of security. Oh no, it's way more sophisticated than that. It has two main systems, and it all depends on where the gatekeepers are stationed. You've got some standing guard on the outer wall, and you've got others operating deep inside the fortress itself.
So what's the difference? Well, it really boils down to location and the type of key they accept. Cell surface receptors, they're embedded right there in the membrane, catching signals that are either too big or too water-loving, what we call hydrophilic, to get through on their own. On the other hand, you have intracellular receptors. They're chilling inside the cell, waiting for special VIP signals that are small and fat-soluble, or hydrophobic, which means they can just slip right through the membrane like they have an all-access pass.
Okay, so let's start with that first line of defense, the gatekeepers posted on the cell surface. These are the guys who greet all the signals that can't just waltz right in. It's time to meet the crew.
This crew is made up of three main players, and each one has a totally different way of getting the job done. First, you have the simple turnstiles, the Ion Channel Receptors. Then you have the internal messengers, the Protein-Coupled Receptors. And finally, you've got the ones that set off a massive chain reaction, the enzyme-linked receptors.
These turnstiles are the most direct of the bunch. When the right key, say a neurotransmitter, binds to the outside of the receptor, the gate just swings open. It creates a tunnel, allowing specific ions to just flood into the cell. This is what drives things like our nerve impulses. Boom. Simple, direct, and incredibly fast.
Next up, we've got the dispatchers. Now, instead of just opening a gate, G-protein-coupled receptors act more like a doorbell. When a ligand presses the button on the outside, the door doesn't open. Instead, it activates a separate messenger inside the cell, a little guy called a G-protein. This G-protein then zips off to alert other parts of the cell, carrying the message deeper. This system is huge. It's involved in everything from your sense of smell to how you react to adrenaline.
And last but not least, in the gate crew, we have the security chiefs. These receptors are really special because the part of them sticking inside the cell is actually an enzyme, or it's linked to one. So when a signal binds on the outside, it flips a switch that turns on this enzyme, which then kicks off a whole cascade of chemical reactions on the inside.
To really see this chain reaction in action, let's look at a classic, the tyrosine kinase receptor. A signal molecule shows up, and it actually brings two of these receptors together. They pair up, and then they activate each other by slapping these little phosphate groups onto their internal tails. This acts like a lit fuse, igniting a whole chain of signals that just explodes throughout the cell.
And this chain reaction isn't just some neat biological trick. No way. It is the fundamental process that drives some of the most important jobs in our entire body, like telling our cells when it's time to divide or how to heal a wound. It's absolutely essential.
Okay, so we've met the guards on the wall. Now, let's sneak past the membrane and meet the inside agents, the receptors that handle the signals that have that all-access pass.
Remember those small hydrophobic signals we talked about? Well, they don't need a gatekeeper on the surface. Why? Because the cell's membrane is also fatty, so it just lets them slide right on through. These are the true VIPs who can walk straight into the cell's command center.
So once it's inside, the ligand finds its partner receptor, which might be floating in the cytoplasm or waiting in the nucleus. They bind together, forming this powerful duo that marches right up to the cell's DNA. And there, it acts like a director, telling the cell which genes to turn on and which to turn off, directly changing the very proteins the cell makes.
So who gets this VIP treatment? We're talking about major players here. Think sex hormones like estrogen and testosterone. Think thyroid hormones, which control your metabolism, and even essential fat-soluble vitamins. These are all signals that need to get right to the heart of the cell's operations to make big changes.
Look, this whole intricate security system is just incredible when it's working properly. But what happens when it fails? Because these gatekeepers are so critical to our health, when security breaks down, the consequences can be absolutely devastating.
One of the scariest security breaches happens when an enemy figures out how to pick the lock. And viruses, well, they are masters of espionage. They very often hijack our own receptors to break into our cells.
I mean, take HIV. It's evolved a key that fits perfectly into the CD4 receptor on our immune cells, using it as a doorway to get in and wreck the whole system. Or look at the SARS-CoV-2 virus that causes COVID-19. It binds to the ACE2 receptor to invade our lung, heart, and kidney cells. They are literally using our own front door against us.
The other huge security breach happens when the gatekeepers themselves go rogue. What if the signal that says grow and divide gets stuck in the on position? Well, you get the uncontrolled growth that we call cancer.
And this is precisely what happens in a cancer called chronic myeloid leukemia, or CML. A genetic mutation causes those tyrosine kinase receptors, the ones in charge of cell division, to go into overdrive. They get stuck on, constantly screaming at the cell to multiply, 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 leading to a flood of cancerous cells.
But here is where the story gets amazing. By understanding this very specific malfunction, scientists designed a master key to jam the broken lock. Drugs called tyrosine kinase inhibitors were developed to specifically block only that faulty receptor. This stops the cancer cells in their tracks, often with very little damage to healthy cells. It was a complete game changer in targeted medicine.
So you see, receptors are so much more than just proteins. They are the control panels for life itself. They're how we communicate, how we grow, how we heal. And as we get better and better at understanding all these different locks, we're also learning how to design our own custom keys. This has already revolutionized cancer treatment, and it really makes you wonder, what other doors are we about to unlock? What's next?